discussing just for 40 minutes or something around the work we have been doing at my institute or my laboratory regarding the animal cloning and genome editing. So, and then that for the prospect for feeding the future population, what we know that farm animals, as well as you can say in a global agriculture, it's very important to provide the sustainable food product. So if we talk about the dairy animals, so particularly everybody needs the milk and milk products. I think it's not a single day from any individual in the world who is not able to have the dairy products either in the form of the milk or even the chai, or you can say paneer, dahi, or everything. So every day every human beings require the dairy product. And to have a dairy products, we have a dairy farming in the country or globally. And to have a dairy farming, so the main target was to um, maintain the animal population with the sustainable production. Production means is a milk production, so that we can generate the different dairy products, including this dahi, paneer, or whatever it may be, cheese or anything. So that's the aim. And to maintain the productivity or the production from the dairy animal, different techniques are used. There's a couple of the techniques, like for example, the breeding techniques, which I am going to talk from the artificial insemination, the assisted reproduction techniques, as well as the cloning. And the future we are working on the genome editing. So this is also very important to reproduce the animal. So because without reproduction there is no production. If the animal is not pregnant or not and deliver a calf, the animal will not give a milk. So it is very, very important to reproduce the animal and reproduce the animal so that the next generation animal will provide the more number of milk, particularly in context to the dairy animal. So my laboratory basically working 80% or 90% work on the buffalo, and the 10%, 20% on the cattle. So regarding the my institution, just I will brief about my institution. So my institution is the National Dairy Research Institute, as the Dr. Gopal is already introduced. So this is our main center at Karnal. So we have two regional stations. One is at the Kolkata and another is the Bangalore. So we have around 150 faculty, which is a stream of the dairy technology, as well as from veterinary field, as well as the basic science scientists. So we have all the different diverse field of the faculty. We have more than 2,000 students. We have undergraduate for the BTech Dairy Technology, BTech Food Science, and BTech Biotechnology, and MSc degree in the different disciplines. So we have around the 17 disciplines from the biotechnology, from the microbiology, biochemistry, dairy technology, physiology, reproduction, and so and so. And also we have a PhDs in this discipline. We have a 5 hostel, so the student can get accommodation there. We have a guest house. Our land is around 500 hectares, and my institution is the largest dairy science university in the country. So in addition to the what are the facilities we have a well-managed dairy farms around the 2,000 animals, which include the cattle, 1,500, 300 buffaloes, 300 goats, 500 buffaloes, and also we have a laboratory animal like mice, rats, and rabbits. We have a bull station to produce the semen, as well as the milk processing plant where we produce the product from the milk. So my discipline where I'm working in the animal biotechnology division or kind of center. We have 8 faculty. We working on the 3 different areas like reproductive biotechnology, where my lab is working. Another area is animal genomics and proteomics, cell science and stem cell biology. The other people are working. We have a couple of good strength of the students, like PhD student, masters, and a couple of the postdoc. This is all about my department. Now, the why we need to have the talk or discuss about these technique, animal cloning and the genome editing technologies. So if you see everybody, I think either the students or faculty, they are agreeing with me is that we are now the most populous country than we are already surpasses the China. And 2060 or 2050, when we are expecting our 100 year of independence, just a moment. हेलो सर, मैं रीजन हूँ सर, एक लेक्चर दे रहा था। हाँ सर बताइए। मैं करता हूँ सर आपको कॉल बैक ना। अच्छा अच्छा सर, मेरे हाँ 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 ठीक। Sorry. So we know that we are our most populous country, so we need to provide them the good nutrition diet in the form of my responsibility as I'm working in the dairy industry. Our responsibility to provide them the good dairy products or to maintain the productivity or high production of animals. So to provide the food to the people as well as to protect the health of the people, because there are so many diseases that have been emerging out from the animals to the human or from humans to animals. So one health aspect is also important. So that's why we have been working on the future technologies. At the moment, these technologies not at the ground level, so not extensively used in the country. But in coming days, this technology will definitely be the game changer to maintain the productivity of an animal. So if you look at the Indian dairy industry, so Indian dairy industry is very huge. So we have a different breeds of the cattle like ghee, Tharparkar, Murrah, and the Sahiwal. We have around the 24% global meat production. Our animal productivity is the lesser as compared to the animal produced in the United States as well as other countries. So 6 to 8 liters of the milk per day our animal producing, as well as in the same time. I think this year also we have been facing the climate change, the heat. Lot of heat was there, so that is also affect the productivity of the animals. So we wanted to maintain the productivity from these animals. So what will be the impact? I think we know that this year there are a lot of the incidences of the disease outbreak, the scarcity of the fodder, and then ultimately it's lead to the low milk production. So we wanted to maintain the milk production from these animals. So if you see how much burden of the dairy industry in India, if you look at the one, just an example of one buffalo, which required every day, it required the 4 kilogram grain fodder, around the 7.5 kilograms of the dry fodder, as well as concentrate with the grains and everything, around the half a kilogram. So this much of the requirement of one buffalo each day, and we have around 109 millions of buffaloes. So to have that, so it required to produce a green fodder, it is required this much liter of water, 1844 liter of water, and this much liter of water for the dry fodder, and this much liter of water, like around the 275 liter of water for the concentrate. So if I look at here, the what is the population of the buffalo? So what I would like to convey from this slide is that we have a huge requirement of water and the land resources to produce the animal. And in the future, our population has been increasing. So what we need to have, because if the population is increasing in case of animal also, they will have a more carbon footprint or more burden of our resources. So we wanted to decrease the number of animal with improved productivity. For example, at the moment the animal is producing 6 to 8 liter of milk. So if we increase by the double or more than that, so our animal number will be reduced, so but productivity will be high. So then it will have a less burden on our resources, either in terms of the land resource, either in terms of the water resource. But the question is that how and when. Now, thing is that these are the techniques which we worked in the farm animal to produce the next generation of offspring. As I mentioned, there is a male and female, the reproduction, the sperms, oocyte, and there are the somatic cells and embryos. So, if it is artificial insemination, it's a natural, similar to the sperms are deposited in the reproductive tract and they fertilize, and then next generation offspring produce. It has been already working in the field condition to produce one. Similarly, IVF, in vitro fertilization, the sperms and oocyte has been like a test tube babies. They produce the animals in the IVF embryos in the laboratory, and then they transfer into the reproductive tract to produce the offspring. Similarly, somatic cell nuclear transfer. I think everybody heard about the Dolly name. That was the first clone animal produced by the somatic cell nuclear transfer. And then again, the embryos are produced, as well as the ETT, where the embryos are collected from the one animal and then transfer and then produce the next offspring. So all these techniques in the country we are working on it at the moment. The artificial insemination as well as the IVF technique has been working in the field condition at the farmers level, and these two techniques, for example, SCNT, is limited to the research laboratories, since the more animals will be produced with that one. What was the impact? Normally, one calf can be produced through the artificial insemination in 1 year from the one cow. But if you go for the OPU IVF from the one cow, in a year it can possible to produce the 15 to 18 cows. Suppose this is high elite animal, so is more number of cows can be produced. Similarly, in case of animal cloning, if we talked about, so more than 100 cows per year can be possible to produce because the somatic cell has been used in this scenario. the cloning, if you look at either it's from the sperms and oocyte, the united
थोड़ा
plant
सा
will
थोड़ा
produce
सा साउंड और वॉइस बढ़ सकता है क्या सर? थोड़ा सा कुछ कुछ लोगों को बोल रहे हैं
moment.
माइक
I
को थोड़ा
mean,
पास रख लेंगे.
आवाज बड़ी है क्या?
हाँ हाँ बढ़िया बढ़िया.
अब वो ठीक है ना?
ठीक है.
Okay. Now, in case of the AI, artificial insemination, embryo transfer, or OPOI, which has the 50% of genetics is coming from the oocyte, not the mother side, and 50% from the father side. But in case of the cloning and the somatic cell has been used, and oocyte, which is in nucleation, there is nucleus free, 100% genetics transfer from the somatic cell. For example, this is an animal. So this animal for here, if we see, so this is making a biological Xerox copies of any individual. So it make an photocopy of an individual to produce the offspring, and which has 100% genetic copy of the particular animal. So in case of buffalo cloning, there are the two techniques: the micromanipulation based and micromanipulation free. So my laboratory in 2009, we produced the world first clone water buffalo at NDRI, and now we have been producing, I think, 20 clone buffaloes in the country, and then we have been analyzing their phenotype, their genotype, their production performance, and then other studies we have been conducted. Now, why we need the cloning technique? As I mentioned, that to produce the elite stock up in a buffaloes. For example, there is a buffalo which is giving the 6,000 liter of milk per lactation. So these buffaloes can be cloned to produce an offspring. Also, for example, conservation of endangered breeds. This is a Chilica breeds which has been in from the Orissa, which has been low number. So these animals can be conserved through the cloning technique. Also, for example, gene modified buffaloes. I will discuss about what is the gene modification and so to have improved milk production and the health trait. So these are the application. So these are the achievements. So just in highlighting here the cloning in my institute, and this is the first clone buffalo here in A is representing the first surviving clone buffalo Garima that is produced the 8 progenies. Also, these are the two clone bulls here in D, and then produce the multiple offspring. Also, the 7 clones from a single individual, which has been producing these. Now they are in semen production. Similarly, the top rank clone bull has been cloned. We have multiple clone copies. Cattle now in last year we produced to the our indigenous technology in case of ghee. So these are the couple of achievements we have been achieving. My laboratory has been did that one. Just I would like to one study. So, is it possible to restore the dead progeny-tested bulls for the breeding? For example, there is a semen is available in the biobank. Can it be recollect or resurrect that genotype back after the death of that particular animal? But the animal was not there, but semen is available. So these studies we published, we did, and we isolated somatic cells from the frozen semen to produce a live calf. And now this bull has been more than the 10 year of age, and then this found the normal growth, and the semen and the progeny is also been produced from this animal. So this was this way it can be helped to resurrect the high genotype animal. In addition to that, in the studies we thought to clone the animals from the distant part. So, for example, this is an Assamese buffaloes. It has been cloned. So this is available in Assam. So somatic cell has been transported to my laboratory. We produce the embryos and then shipped it to into the another field conditions around the 90 kilometers away from the laboratory. And this is a calf has been brought from the recipient of Murrah. So this calf is now, I think, is producing semen, and then several parameters we studied like the normal physiology, the biochemistry, serum hematology, the chromosome number, genetic diseases, and the producing semen. CASA parameter, this computer assisted the semen analysis we did. Also, we did the IVF-based fertility to make sure is that this animal is fertile. If you see here is a normal mounting, and this is also one of the pride animal we produced. In addition to that, we thought to have generate the next sequencing data about the mRNA and miRNA between the clone bulls and the non-clone bulls. What is there any effect or not? So we did not find any difference between the transcript abundance between the clone and the non-clone spermatozoa. Similarly, the IVF-based fertility and AI-based fertility has been done. Also, we did the 300 artificial insemination using the clone bull semen, which has the normal consumption rate, 40 to 50% of the consumption rate. This is a journey of buffalo cloning in India. So, just in summarize here, in 2009, we produced the first clone at NDRI, my institution. Our embryo production rate this much, our live birth rate is around 6 to 8%, though we have a challenge about the pregnancy rate of 20 to 30%. So, different clones are produced, around 23 clones, of which majorly they have now been from the breeding bulls. Also, what we have been did from the last decade, work we have been done, the clones that have attained the maturity have the normal growth, health, fertility, and the production. So this is why we require, as I mentioned, we need to produce higher productive animal, and that animals to have with the more number of milk. So if you look at the Indian scenario, we have around the breedable female population 123 million. Out of that, in buffalo is 54 million. So, but AI coverage is at the present, artificial insemination coverage is at 30%. So, we wanted to increase by 100%. In that sequence, we require the huge number of semen doses. So, that's why we have been majorly using the breeding bulls to be a clone, and that can be helped to satisfy demand of quality bulls and semen doses for the breeding program, so that the more animals can be a clone. So this is how it can be used. So for example, there is a bull. If you go for the classical breeding, this much next generation calves can be produced. If you go for the declared proven bull, if you have multiple clone copies, more number of progenies can be generated on the next calf. So to have what is our plan to avoid the inbreeding and to maintain diversity. For example, the bull is at the in my state here in Hisar or here in Karnal at Haryana or Hisar. So this bull can be goes to the different states, for example, Andhra Pradesh, Telangana, Karnataka, Andhra Pradesh, and Odisha, and the Bihar, the different part. Also, since we have a good buffalo germplasm, it can be exported to the different parts of the world. So, export to the different Asian countries. Also, it is not recommended not to use the bull in the same state as we saw. For example, bull in Hisar should not be used. Also, it should not be used for more than the 2 years, 3 years. Also, it can be possible to export these animals or the semen into the Latin American countries where the buffalo population is available. Now, why we clone the elite animal? This was a clone we produced in 2021. Now we started to doing the OP of this animal in 2024, and we expected that this animal can be used to produce more future breeding bulls as the oocyte donor for embryo production. So next generation offspring, higher milk production offspring can be produced. Also, since it is very high, so it has been used the therapeutic protein productions in the milk. So if anybody interested to go through the what work we have been done, so a couple of publications here. I would like to mention the review articles from my laboratory. The cloning is been available, as well as this book chapters. As a couple of research articles, you can search the PubMed to understand what work we have been done. Now coming into the this is all about the cloning, what we have been done. Our primary interest of the cloning is to produce the breeding bulls so that the semen can be used to upgrade the future populations, so we have the more productive animal. Another the work we have been started from last couple of years about the genome editing. So gene phenotype, and that ultimately can be modified through the genome editing. So DNA will be, we know that everything is the DNA is considered to be a major messenger available in our body in the mammals, so which can be major responsible of phenotype. In case of dairy animal, is milk and meat if you consider, and that is industry demand, like dairy industry, if you say the milk. So in the genome editing, it is possible to alter the any trait, and that ultimately what we require have high milk demand or disease resistance or meat demand, whatever it may be, so we can fulfill with the industry demand. So what is actually the genome editing? I think there are a couple of talks have already been done on the genome editing by in the dialogue series, which is organized by Dr. Gopal and his team. So I will not go into the detail, but just in summarize or brief about that. It's a CRISPR technique which has make a cut by this is a double stranded cut or single stranded cut in the DNA level, and if you have not providing the donor template, so it will goes to the non-homologous end joining pathway that lead to the insertion and deletion, and that lead to the knocking out of a gene. If we provide the donor template, so the new DNA can be constructed through the homology directed repair. So this mechanism has been available in mammals, so especially eukaryotic cell. But basically, these technique which has been adapted from a virus, there's a bacterial immunity. When there is a virus particle go into the sequence, this sequence, the spacer sequence integrated into the bacterial genome. And when it has been transcribed, it's the RNA and with the Cas9 protein complex. When the second time the bacteriophage infect, that time they make the double stranded cut again. So there is a bacteria is immune to the infection by the bacteriophages through this CRISPR technique, which was subsequently employed in the case of eukaryotic cells, particularly mammals, including plants, humans, farm animals, and everywhere. So what is actually the Cas9 allow the cutting of these genomic DNA using a gRNA. So it is possible to add, delete, and modify the any nucleotide. Uh, as compared to there are other genome editing tools, but CRISPR has been more simple and efficiently, and that's why it has been replaced to most of the other genome editing tool like the ZNF and Talon. And this technology has been in case of dairy animal, if I can say, the more productive animals as well as disease-resistant animals, it has been done. So why we need in case of, for example, in dairy animals, if we say why we have been using, suppose they're favorable in terms of the milk production. So it takes 8 to 10 generations. So 8 to 10 generations in dairy animals means at least of 20 years of time to migrate into the favorable population. But if you through the genome editing, it is one generation. So like in case of zygote, one generation you can have a favorable animal. Like example, one generation in 3 years. Within 3 year period of time, you can generate the favorable population. So what was an application? So to improve the milk production, so we mitigate the demand. For example, milk yield is more than the 10,000 liters. So meat yield, example, if we case of 500 kilograms in the 5 months. A disease resistance animal, so they should not have a diseases like FMD, the foot and mouth disease is very pathogenic disease, an economic disease in the dairy animal, as well as brucella, which is also and zoonotic disease, which can cause both human as well as animal. Also welfare of animal like hornless cattle and coat color and other things. So this can be done. So if I look at the application of genome editing in dairy, in case of animals, all over the other animals also is a milk and meat production, where it can be modified. The milk trade, wool and hair production can be also be modified. The welfare of the animal, like pole cattle from the pole to the horn cattle to pole cattle, the diluted coat color for the climate resilience, so from the light to dark, developed through the CRISPR. So it is one. Xenotransplantation is very, very important now. The emerging field that many people is talking about. How can we use these the pigs as an animal where the human organ can be grown so that they can mitigate the demand of the organs for the human transplantation. Also, the disease resistance. So disease resistant animal like the animals which is not getting the infections. So these are the disease animals can be produced. Now coming into the my laboratory is working on the couple of disease, a couple of targets. So here just I will highlight these buffalo, our work in the milk production traits, particularly alternating days one. So if we talk about the India's white revolution means milk production cannot be imagined because of the buffalo. And we know that every year we are producing 240 metric ton of milk. It is a top ranked first and production in the world. But that is majorly is because of the buffalo is around 45% of the milk produced in the country is contributed by buffalo. But what happened is milk, we know the consider is a complete diet. But in addition to that, there is a beta lactoglobulin. This is another one allergen protein present in the milk. So couple of human, particularly newborn, one babies, they have been allergens to the milk. So to avoid that, so we thought to have the knocking out of this a beta allergen from the genome of an buffalo. So we worked on the direction. So this is what we have been achieved is that we select the buffaloes, the CRISPR RNAs has been designed and transfected into the cell and generate the clonal population, and then ultimately we produce the knockout BLG embryos, and these embryos we develop and then transfer into the recipient animal to establish pregnancies. And we hope this year we will produce the buffaloes that has been BLG knockout clone animals. So the efforts are continuing underway to develop buffaloes that can produce a BLG free milk. So not yet. So it is particularly what we have been tailored the milk, the crafting allergen free options. Just to sum up of this, what we have been done. So it is a very well established the buffalo cloning technique for the breed improvement and gene editing. What we have been working on it. Also, we have been working on the MSTN and BLG gene editing, which is under progress. We know that we have the highest opportunities in the farm animal, but in addition to that, working with the animal is really challenging task. It's not like working with the cell culture or in the laboratory animal. It's very tedious and laborious, and to have to manage the animals is also the a couple. So I would like to acknowledge the different funding agencies who has been supported our work, like Indian Council of Agricultural Research, Bill Gates and Melinda Foundation, which has been supported. My two institutions like NDRI, where I'm currently in my previous institution, CIRB, Department of Biotechnology, who has supported the project, as well as the couple of projects like CRISPR, mice cutting project in Taylor Mill, as well as my team members at the farm team, as well as the laboratory team. We have a laboratory team as well as the farm teams also at the different institute. Thank you very much, and I would like to happy to take the couple of
Thank
questions we
you.
have.
Thank you. Yes, yes. Video is
Okay.
coming. So,
Yeah, yeah. So this is a 6 minute video. So how
yes.
we produce the animals so they can understand. So these are the immature oocyte. So there's oocyte from the animal. So these oocytes are collected. Then after the maturation, how the oocytes look like. Okay, these are the mature oocyte, and then we remove the cumulus cell and then make them the denudate. And then make them the zona free, so there is a no zona pellucida around. And if you see here, this is a protrusion cone where the DNA content is localized. So if you see here, this is a bright spot in between here. So this is a, yeah, this is a bright spot which contains the genetic material metaphase 2. And then it has been selected the oocyte. Then it has been aligning the oocyte in the bisection dish. And this is a micro blade we use for the bisecting the oocyte. And then under the stereo zoom microscope, this oocyte has been enucleated into the two parts. One part which contains the DNA material, another part is the enucleated oocyte. These are the fibroblast cultures. So there is a monolayer culture. And then it has been trypsinized to make them detach from the surface. This is a rolling of the one cell. If you see here, this is a somatic cell and this is enucleotide rolling over there. This is a pairing we made. So one somatic cell and the one enucleated oocyte pairing has been done, and then this pairing, when under the again in the stereo zoom microscope with the very sophisticated machine, fusion machine, so they have been fused like hybridoma culture. So they have been fused, and then they have been mixing up their cytoplasm. So if you see here, lead how the cytoplasm of the two oocyte and the genome has been mixed together. So from the dumbbell shape, it will be now completely rounded one. So if you see here, there are the different stages of the fusion. One is completely round, another one. And then it has to be chemically activated to them to make the cyclicity of the oocyte to reach to the from metaphase second to cyclic oocyte so that they can start division. Like this is a wall culture. So these are cultured into the laboratory to get them into these blastocyst stage, like 2 cell stage. This is the 4 cell stage, and then it will reach to the blastocyst after some time. After the days of the 7 to 8 days, they reach to the blastocyst, and these blastocysts This is again the 8 cell stage, and this is the blastocyst stage where we have inner cell mass and profectodon, and these are embryos are then transferred into the recipient animal. So I will show this is again the flat surface system. Another approach. This is a compact morula, and then it will read to this is if you see these are the very beautiful blastocysts which have been generated in one trial. So many blastocysts. So each animal will receive the one blastocyst. It is called you can say the babies of 6 days or 7 days. These are 6 7 days babies. This is again a blastocyst picture which is in glass oviduct, another system which is in oviductal kind of culture to provide them in that situation. This is a staining. So if you see the red color, which indicated the inner cell mass cells, which is gives rise to the fetus, embryo fetus, and then once it these embryos they have so this is been transferred. This has been transferred into the recipient animal. If you see here, from the one hand in the rectum, another hand into the vagina, and then this gun will goes into the reproductive tract of an animal when it has been deposited on the uh, site where the corpus luteum is present. And once it has been transferred, we did the ultrasonography to detect the pregnancy. So if there is an animal is pregnant or not. So this is a pregnancy diagnosis to the probe. So if you see here, this is the actually fetus, which is this is a fetus which is a pregnant fetus of around the 40 days. So this way we will produce the embryos and then uh, transferring them into the recipient animal to establish the pregnancy.
Yes.
Hello.
Yes, yes. बहुत बढ़िया वीडियो.